Hi guys, welcome back to another CXE revision session. I'm your host, MISSBIOCHEM'S. So today we'll be reviewing paper 2 of the May, June 2016 CXE Biology past paper. Let us begin. So in section A, answer all the questions in this section. Write your answers in the space provided in this booklet. So question 1A. Students investigate the effect of temperature on the rate of a reaction of the enzyme catalase. They use crushed potato as the source of the enzyme and place 5 mg into each of the 7 test tubes. They add 3 centimeter cube of 1% hydrogen peroxide to each test tube and place them into water baths kept at the following temperatures. So 0 degrees Celsius, 10 degrees Celsius, 20 degrees Celsius, 30 degrees Celsius, 40 degrees Celsius, 50 degrees Celsius, and 60 degrees Celsius. They count the number of bubbles released per minute from each test tube and record the data in Table 1. So Table 1, effect of temperature on catalase activity. So now we have a table. So we have the test tube. Remember they told us 1 to 7. And then we have the temperature recorded and also the number of bubbles produced per minute. So question I. Plot a graph on the grid provided on page 5 to show the relationship between the number of oxygen bubbles released and temperature. So now we're going to plot a graph with these values that were collected, and they told us to do it on page 5. Going to page 5, which is this. I've already plotted my graph. So it looks like this. So this is my graph. So all I did was figure out what should go on the y-axis and what should be on the x-axis. So based on what they said, plot a graph on the grid provided on page 5 to show the relationship between the number of oxygen bubbles released and temperature. So by reading this, I now know that the number of oxygen bubbles released should be on the y-axis, and then the number of temperature, and then the temperature rather, would be on the x-axis. So now I have number of bubbles per minute on the y-axis, temperature on the x-axis. So all I have to do is fill in the data. So normally when you're trying to create a graph, whatever information or data that they give us first is normally on the y-axis, and then the one that comes after is normally on the x-axis. So to plot our graph, in the first test tube, the temperature is 0 and the number of bubbles is 0. So if we go back to the graph, temperature is 0 and the number of bubbles is 0. So the first point would be here where it's 0 meets. Then the next one, for the second test tube, temperature is 10 degrees Celsius and the number of bubbles produced is 1. So temperature is 10, so temperature is on the x-axis, temperature is 10, number of bubbles produced is 1. I'm going to show you guys one more, but you should be able to plot your graph on your own. So the third one is temperature is 20 degrees Celsius and the number of bubbles is 5. Going back, temperature is 20, look at the temperature sign, it's 20. Number of bubbles is 5. And we would have our next point here. Another thing that I forgot to tell you guys is whenever you are making a scale for your graph, you have to look at the information you have gotten in your table to know what your scale will be like. So in the temperature section, we realize that we have 0, 10, 20, 30, 40, 50, 60. So we're already going up by 10. So that's a good scale to use. So that's why I use that same scale on my graph. So 0, 10, 20, 30, 40, 50, 60, 70. All right. And then on my y-axis where I have my number of bubbles, so the values that I'm seeing are 0, 1, 5, 10, 15, 1, 0. So I have to come up with a way in which I can get all of these units or all of these numbers. So I counted by 1 and I ended up at 15 because remember 15 is the highest number that we got in the table. So I count by 1, 1, 2, 3, 4, 5, 6, 7, all the way to 15. Another thing now is to create your scale. So I have a scale. So on my y-axis, I've already told you guys that I'm counting by 1, but it's 1 centimeter to 1 unit because it's 1 square. So it's 1 unit. As you can see, 1 square, 1 unit. But on the x-axis now, we're counting by 10. So we're increasing by increments of 10, going up by 10. So it's 10 centimeters now. I have 2 units, and that's because we utilize 2 square. 1, 2. So that's why it's 2 units. And also remember to give your graph a title. So I have a graph showing the relationship between the number of oxygen bubble release against temperature. And that should be good enough. Or you can always go back to question I, which I got my information from, and say something like a graph showing the relationship between the number of oxygen bubbles released per temperature. Or even the title of the table could give you a graph title. So a graph showing the effect of temperature on catalase activity. And you should be good. So the next question, which was here. So from your graph, determine the optimum temperature for the enzyme. So remember, the optimum temperature would be the highest point or the highest peak in which that enzyme, which is catalase, will work best. And as you can see, the highest point would be here. This is where we have it. So it's 15 bubbles to the temperature, which is 40. So the optimum temperature now is going to be 40 degrees Celsius, and that is why we have that on the line. Moving on. 3. Explain the shape of the graph at each of the following temperature ranges. So we have 0 to 10. So we have to explain what is happening. So looking at the graph, 0 to 10, 0 to 10, we can see that the temperature just starts to increase, and that's why we have like a small amount of bubble, which is just 1 bubble. So we can explain something along that line. So I have the temperature is low, so there is a low enzyme reaction, hence small amount of bubbles seen. As we can see, it's 10 degrees, 0 to 10. So the temperature is definitely low, so not a lot of reaction is going on, and that's why we're not getting a large amount of bubbles. So at 11 to 40 degrees, let us look at the graph. So at 11 to 40, so 11 would be after 10 to 40, we realize that there is a drastic increase in the temperature as well as the number of bubbles, because at 40 we have 15 bubbles, so there's a lot of bubbles there, and the temperature increases from 10 to 40 degrees. So now what we can say is there is a significant increase in temperature. More enzyme reaction happens as the increase in temperature speeds up the enzyme reaction or the enzyme activity. So the increased temperature that we have would just ensure that the enzyme is working best. So the enzyme is doing a lot of work. So I'm moving on to the next one, which is 40 degrees Celsius and above. So we have the greater than sign. So greater than 40 degrees Celsius. So now greater than 40, we know that at 40, the enzyme has reached its optimum temperature, which that enzyme catalase will work best. So above 40, now we realize that there is a decrease in temperature because it's going down. So what we can say is the enzyme the enzyme reached its optimum temperature and is no longer effective, that is, the substrate will not be able to fit into the active site. So once the enzyme has reached that optimum temperature, it will no longer work. So the enzyme has reached that temperature, so it's no longer working properly or effectively. Hence, there is a decrease in the number of bubbles being produced. IV. Write a conclusion that the students could draw from their results. So it can be concluded that enzymes are affected by temperature, and the enzyme catalase work best at 40 degrees Celsius. So this is the information. This should get you your 1 mark. So please remember, whenever you're writing a conclusion, it comes off your aim. So whatever the aim is, your conclusion should come off it. If you were to read through the information that they provide you with, chances are you would see some information there. You may even spot your aim. So let us read this one more time. So students investigate the effect of temperature on the rate of the reaction of the enzyme catalase. So if you remember how to do a lab, you would have realized that the aim is right here. Investigate the effect of temperature on the rate of reaction of the enzyme catalase. To be more specific, to investigate the effect of temperature on the rate of reaction of the enzyme catalase. So remember, normally whenever we're given an aim is to investigate, to determine, to analyze, all of that. So if you see here, you would have realized that this is your aim, and hence you must conclude based on this aim that is there. So your conclusion should be something like, the effect of temperature on the rate of enzyme reaction was investigated and it was successful. And you could give them more information based on what you got. You could tell them the optimum temperature like I did. And you should be okay. It's 1 mark. Moving on. So just one reason why living organisms need to maintain a constant internal temperature. So remember, homeostasis is the maintenance of the constant internal environment of the body. So now, if the internal temperature must be constant, or otherwise all the reaction needed in living cells may be disrupted, or the body will not be able to catalyze it, which is pretty simple. If your internal temperature is not being maintained, then certain reactions will not be happening and certain things will be disrupted in the cells, and that's why we need balance. VI. Describe 2 precautions that the students should take when doing this experiment. So 1, ensure that gloves are worn. 2, ensure the temperature are correct in the water bath. So please remember that each water bath had a different temperature. So if they're not correct, meaning that it's higher or below, then it can affect the reaction. It can affect the number of bubbles we see. So 3, ensure the bubbles are counted properly. As usual, we have to ensure that it is counted properly so we can get the most accurate result. Then 4, ensure the results are taken. So ensure the results taken are accurate. So I'm missing results here, but that should be there. Ensure the results taken are accurate. So let us see if we can put results here. So that's R, E, S, U, L, T. So ensure the results taken are accurate. So any one of these combinations should be okay to give you 2 marks. You don't have to put all of them. I'm just giving you guys some extras. So VI, how could this experiment be modified to investigate the effect of pH on enzyme activity? So now you have to incorporate pH in there. So for this lab, it is actually pretty easy to incorporate pH. All you have to do is figure out a way to test for pH, and you would have known that you can use a pH meter or a litmus paper. So all you have to do, or all you have to say, is that you're going to incorporate a pH meter or a litmus paper into the experiment. So now you have to tell them how are you going to use the pH meter, and then this is what I have written down for us. So the pH meter or litmus paper can be incorporated. The students can take the pH of the reaction before it is placed in the water bath and take it again after the bubbles per minute are counted to investigate if pH affects enzyme. And that should be pretty simple. So all they have to do is take the pH of the reaction before, and then whenever the reaction starts, that would be when you put it in the water bath. So temperature speeds up a reaction, yes. So that would be when you put it in the water bath. So after you counted your bubbles, because you don't want to put your pH meter or your litmus paper in there because it can affect the amount of bubbles that will be produced. So it would be best if you do it after you have counted your bubbles, just to see if the temperature has, just to see if the pH has changed from before and after. Another thing you can do, you don't have to take it before, you could just take it after, whenever the reaction has completed, just to see if enzyme affect the temperature. Just to see if the pH is going to affect the enzyme reaction rather. So B, name 2 enzymes that function in protein digestion and describe the role of each. So I have pepsin and trypsin. So this is found in the stomach and break down protein into smaller peptides, and that's a pepsin. And then trypsin, this is found in the intestine. It continues the breakdown that have begun in the stomach of protein into smaller peptides by breaking the peptide bonds. So we also have chymotrypsin, which is also found in the intestine and also breaks down protein. And we have another enzyme that is in the stomach, but it doesn't necessarily break down protein. It breaks down milk protein or dairy products. So milk protein, and this is called renin. So renin is in the stomach. It breaks down milk protein. But pepsin, trypsin, and chymotrypsin, they all break down regular protein. Moving on. C. Before food can be chemically digested by enzyme, it must first be mechanically broken down in the alimentary canal. So I name 2 parts of the alimentary canal that are responsible for the mechanical digestion of food. The first thing that should come to your mind is the teeth. As you can see, I have the mouth and the teeth, and it is responsible for mechanical digestion. It tears and breaks and chews food. Then I have the esophagus, and the action that helps with the mechanical digestion is peristalsis, in which whenever the food that we eat are churned up, form the bolus, when it's pushed down into the esophagus, it will contract and relax to force the food or push the food further down into the stomach, and it is called peristalsis. So the stomach now also aids in the mechanical digestion of food, as it's a muscular wall that relaxes and also contracts to turn the food as it arrives from the esophagus. So any one of these 2 should go. So any 2 out of these 3 should go. So 2, give 2 reasons why mechanical digestion of food is important. So 1, mechanical digestion breaks down food into smaller pieces so that chemical digestion can take place. And then 2, mechanical digestion helps to move food along the alimentary canal. So 3, explain the significance of chemical digestion of food. Chemical digestion allows the body to absorb food that we eat. 2, chemical digestion utilizes chemicals, example, enzymes, to break food materials down into smaller components. So utilize enzymes that's going to break food / materials down into smaller components. Question 2AI. In the box provided below, draw a food web consisting of the following organisms. So we have algae, small fish, tadpole, water snake, egret, eagle. So whenever I'm drawing a food web, the first thing I try to identify is which organism is going to be my producer. So if you're reading through your list of names that you've gotten and you realize that there's not a plant there, then you have to think about a microscopic organism being your producers. These organisms are normally some form of bacteria, so some microorganism rather. And these organisms are also expected to be photosynthetic, so they can photosynthesize. So we have the algae, the small fish, the tadpole, the water snake, the egret, and the eagle. So the only organism here that can be photosynthetic is the algae. So now I'll have the algae at the bottom, or starting my food web rather. And then the next thing I normally do is to figure out which organism would be my top carnivore, or would be the most carnivorous, or would be the predator. So this organism should be at the top of my food web. And if you remembered the list, it would have been the eagle. So now we identify the organism at the top and the organism at the bottom. So all we have to do now is to fill in the spaces. So the next thing that we need to do is find an organism that is small enough to be eaten by something, but still large enough to eat the algae. So in our list, we would have the small fish, the tadpole, the water snake, and the egret. So we'd realize that the small fish would be small enough to be eaten by something, but yet large enough to eat the algae. And it's the same thing for the tadpole, small enough to be eaten by something, yet large enough to eat the algae. So now we would draw a line from algae to tadpole, a line from algae to small fish. So the next thing we need to know is, so now we'd be left with the water snake and the egret. So we have to figure out if we want them, if they would be in the same level, trophic level, or a different trophic level. So now we have the water snake and the egret. So now I put them in the same trophic level. So we have the egret, because the egret can eat the tadpole. Definitely remember, egrets are the white birds that normally perch on top of the water to eat fish, and the egret can also eat the small fish. So now, although the egret can be able to eat the water snake, it's not one of their main dish. So that's why I have them in the same trophic level. But definitely small fish is one of the egret's main dish, or fish in general. And they can also eat the tadpole. And that's why I have an arrow from the tadpole to the egret, arrow from the small fish to the egret. So the water snake they can also eat the tadpole and also eat the small fish. And that's why we have arrows from both of them going to it. So now the eagle is a predator, one of the top predators in the food web. So it can also eat the egret and also eat the water snake. So we have completed our food web now. So the next thing, what is the role of the organism at the start of the food web, which is the producers? So the organism at the start of the food web provide the other organisms with energy, so that's their role. So name the organism which performs each of the following functions in the food web drawn in AI on page 10. So A, both as a prey and a predator. So we have to find organisms that are a prey. That means they can be eaten, and a predator, that means that they can eat another organism. So both as a prey and a predator. So we have the egret, the water snake. They're both a prey because the eagle eat them, and a predator because they can eat the tadpole and the small fish. And it's the same thing for the tadpole and the small fish. They are both prey and predator. They can eat the algae and they can also be eaten. So any combination of those four should be good. B, the primary consumer. So small fish / the tadpole, that's what I have. So the primary consumer is the first organism to eat the producers, and that's why it's the small fish and the tadpole, because the producer is the algae. So the first organism to eat it is the primary consumer. So the next one is the secondary consumer. So the secondary consumer would be the egret and the water snake, because there is a second organism to eat the other consumers, which would have been the primary consumers. So it would have producers, primary consumers, secondary consumers. And then the tertiary or top carnivore, which we already spoke about, is the eagle. So now from your food web in AI, draw a food chain consisting of 3 organisms. So I have algae, small fish, egret. So you don't have to make that choice. You could also go a different route. So algae, small fish, water snake. Could also go algae, small fish, eagle. Could also go algae, tadpole, water snake, algae, tadpole, egret, algae, tadpole, eagle. So remember, it has to be 3 organisms though. So any one of those combinations should be okay. So what is the role of the organism to which the arrow points in the food chain? The organisms are consumers that cycle energy. So that's their role. They're going to consume it and they're going to cycle it. So the energy is going to go from one organism to the next because it's a cycle. So explain why energy flows in one direction in the food chain. Energy is unidirectional, and it moves from producers to consumers and cannot move from consumers back to producers, mainly because you would have eaten your producers. But yeah, so it's unidirectional. Unidirectional means it only goes in one direction, it doesn't go back. And that's why I have here from producers to consumers and not the other way around, not from consumers to producers. And as I mentioned, that's mainly because we normally eat our producers, so we can't give them back the energy. So list 3 features of an environment which supports living organism. So they're mainly talking about biotic and abiotic factors. Any combination of those 3 should be okay. So I have climatic factors such as light, temperature, rainfall, wind, and sunlight. Then the ecosystem, which is self-sustaining system with organisms interacting with each other and the environment. And I also have other living organisms such as predators, parasites, competitors, pathogens. So they also help to support living organisms. 3. Define each of the following terms. So we have stimulus, receptor, effector. So a stimulus is a change in the environment, while the receptor, a part of an organism which detects the stimulus. Example, sense organs in animals or the tips of roots and shoots in plants. The effector, part of an organism which responds to the stimulus, muscles and glands in animals, the region just behind the tips of roots and shoots in plants. So B, figure 1 is a diagram of a section through the human eye. Label each of the following parts of the eye in figure 1. So I've already labeled the pupil, which is here, the ciliary muscle here, the retina right here, the optic nerve here. So why is X called the blind spot? And X is right here. So X is called a blind spot. So it's the point where the optic nerve leaves the eye and no light sensitive cells are there. So that's why it's called a blind spot. So no light sensitive cells, meaning that no cells that are in the blind spot can detect light. So it's blind. Blindness has to do with seeing, darkness, because there's no cells there to detect light. Then it's like in complete darkness. So it's the blind spot. Question C. A boy looked up from reading his book to see a helicopter flying in the distance. Account for the changes that would occur in the lens of both of his eyes to enable him to see the helicopter. So the cornea bends light rays from distant object almost parallel. When focusing on distant object, the ciliary muscle relaxes. This pulls the suspensory ligaments tight, which makes the lens flatten or less convex, the shape refracts the light less and the image is focused sharply on the retina. So D, figure 2 shows two eyes labeled A and B under two different light conditions. So the question, which of the eyes in figure 2, A or B, shows the appearance of an eye exposed to dim light? Explain your answer. So A would show the appearance of the eye exposed to dim light. So in dim light, the pupil expands to take advantage of as much light as possible to enter the eye. The radial muscle contracts while the ciliary muscle relaxes, pulling the iris back so the pupil open. In layman's term, in dim light or in the dark, the pupil will expand as the pupil wants to take in more light, so it will get bigger. And the bigger it gets, the more light it can take in to see what is going on in the area. And as a result, once the pupil is getting bigger, the iris will get smaller. So now E, why is it important for the eye to respond when exposed to bright light? So when very bright light hits the retina, it can overstimulate the retinal cells, which release too many chemical signals, which can damage the back of the eye. Section B. Answer all questions in this section. Write your answers in the space provided in this booklet. So we have 4A. In the box below, draw a labeled diagram of the female reproductive system using an arrow. Trace the path of the spermatozoan or the sperm would have to take to fertilize an ovum, which is the egg. So I've already drawn the female reproductive system. I've given it a title, a diagram showing the female reproductive system. So now I have orange representing the sperm direction. So the sperm would enter the vagina, pass through the cervix, then enter the uterus. From the uterus, it would go into the fallopian tube or the oviduct, where it would meet an egg. If an egg is there, it will fertilize the egg. And that's the strip of the sperm into the female reproductive system. Question B. A young couple wants to choose between two contraceptive methods, barrier or surgical. Discuss why the male prefer the barrier method rather than the surgical method of contraception. Your answer should include 2 advantages of the barrier method and 3 disadvantages of the surgical method. So now the advantages. The barrier method is cheap and convenient and effective. So it's cheap, convenient, and effective. 2, it also protects against STI and STDs. So disadvantages. Disadvantage of the surgical method is that it requires surgery, which can have a risk of surgical complications. 2, it is relatively irreversible, especially when the couple decides to start a family, and it can be difficult for them. Then 3, it does not protect against STIs and STDs. C, a biologist discovers that the flower of a species of plant have no male reproductive parts. Suggest one method by which these plants produce offspring and 3 advantages of this method. So the method is basically asexual reproduction. If you want to be specific, you would speak about vegetative propagation. So now advantages. So offspring can be produced continuously and therefore quickly. Large numbers of offspring can be produced, that's 2. And then if the parent is of superior quality, all the offsprings will also be of superior quality. That would make it 3. So next question, 5A. With the aid of a chemical equation, explain what is meant by the term photosynthesis and discuss how a cotyledonous leaf is adapted to capture maximum sunlight and carbon dioxide for the process. So photosynthesis is the process by which green plants make carbohydrates by combining carbon dioxide and water using sunlight energy absorbed by the chlorophyll in the chloroplast. So the equation is 6CO2 plus 6H2O, conditions, sunlight, and chlorophyll to give us C6H12O6 plus 6O2. And that would have been the equation. So the adaptations now. So the laminae are broad and flat and provides a large surface area to absorb sunlight and carbon dioxide. That's one. Two, laminae are thin and enable sunlight and carbon dioxide to reach all the cells rapidly. Laminae lie at 90 degrees to sunlight and maximize their exposure to sunlight. Laminae are spaced around the stem and maximizes their exposure to sunlight. Stomata in lower epidermis allow carbon dioxide to diffuse into the leaf and oxygen to diffuse out. Air spaces between the spongy mesophyll cells allow carbon dioxide and oxygen to diffuse to and from cells. The chloroplasts can move to the top of the palisade cells and arrange themselves broadside to the light, especially in dim light, to maximize the amount of light captured. The xylem vessels and phloem sieve tubes carry water and minerals to leaf cells and carry soluble products away from leaves, which is photosynthesis respectively. Wax cuticle prevents the loss of water needed for photosynthesis. So it is 11 marks. So I have 1, 2, 3, 4, 5, 6, 7, 8, 9 adaptations. So that's 9 marks plus 1 for the equation, that's 10, and 1 for the definition, that's 11. So that's how you get your 11 marks. So B, outline how decomposers assist in the cycling of carbon dioxide to make it available to green plants. So waste materials from living organisms like urine and feces and their dead bodies, so in bracket all organism dies, are used as food sources for decomposers. Decomposers are bacteria, fungi, and worms. Bacteria can break down most type of organic matter and is a significant decomposers. Fungi are primary decomposers in the forest where they break down fallen trees and other woody organisms. Worms are scavengers that hasten bacterial decay by breaking an organism down so the nutrients are more available to bacteria. Carbon atoms then becomes incorporated into the bodies of the decomposers, and respiration of the decomposers releases carbon dioxide into the atmosphere. So as you can realize, I've basically told you guys how all of these different decomposers work together, but for the most part we all know that bacteria is one of the main decomposers, and as such it will break down whatever waste, whether dead bodies or urine or feces, that is in their present environment, in their ecosystem. And when they do that, they're gonna incorporate the carbon atoms into their bodies, and whenever they respire, that carbon atom will be released into the atmosphere back as carbon dioxide. So it will be released back into the carbon cycle. So saying all of that you should be able to get your 4 marks. So now question 6. State 2 causes of anemia and describe 2 signs and symptoms of the disease. So the cause, so we have reduction in the number of red blood cells / hemoglobin production. It reduces the oxygen carrying capacity of the blood. And then the symptoms, weakness, fatigue, shortness of breath, increased heartbeat, pale appearance, reduced oxygen carrying capacity. So any 2 out of these 4 signs and symptoms should be getting you your marks. So now B. Sickle cell anemia is caused by the presence of the recessive allele. Persons who are heterozygous and carry sickle cell allele are more resistant to a type of severe malaria than persons who are homozygous for the normal hemoglobin allele. So state what is meant by each of the following terms. So homozygous, if the allele of a gene are the same, example A and A for normal, or small a and small a for the sickle cell or albino gene. So if I'm homozygous, I can either be homozygous dominant or homozygous recessive, and that's why I have capital A. Capital A would represent dominant, and common A would represent recessive. So for homozygous, they have to have the same gene. So they have to have either 2 capital or 2 common, have to be the same gene. So now if we're heterozygous now, if the alleles are different, example, capital A and common A for sickle cell carriers, meaning that you have 2 different alleles. One will be capital and one would be common. And that would mean that you're heterozygous. So now 2, with the aid of a genetic diagram, show how a man and a woman who do not have sickle cell anemia can have a child who has the disease. So for the person to have a child who have the disease, then guess what? The parents must be carriers. They must carry the sickle cell disease, so they must be carriers. So the parental phenotype traits, so the parental genotype, so the phenotype will be the trait because they are only the carriers, and we call the persons who are carriers, they have the trait, the sickle cell trait. So the parental genotype would be HBNS and HBN. HB itself has to do with the hemoglobin, and the S would represent the sickle cell. And that would be the genotype of both parents because they carry the gene, but they don't express it. It's not shown. And that's why they can have a child who has sickle cell disease. So now we have to separate it. So this is up on its square. So we have HBN and HBS because we separated the HBNS. So we have HBN, HBS, same thing. So HBN times HBN will give us HBNN, which is a normal person. And then HBN here times HBS would give us HBNS. Then moving on to the next one, HBS times HBN, HBNS, and then HBS times HBS would give us HBSS. So this child here, this one, would be the person who has the disease. These two would be the carriers. And then this one would be a normal child. So the genotype HBNN, normal as we mentioned. HBNS is the trait. And then HBSS is the sickle cell anemia, so the person have the disease. So I did it twice. I did it doing the Punnett square, and I also did it using the genetic crossing. So it's the same thing. The parental phenotype is the trait, and then the genotype is HBNS. HBNS are both parents. So the gametes are the N and the S that would give the child whether they're going to get sickle cell or they're going to be normal. So that's why we have them separated and circled. So now N times N would give us somebody with HBNN. Next one, N times S would give us a person with HBNS. If we use the next one now, S times N would give us somebody with HBNS. And then S times S would give us a person with HBSS, which would have the sickle cell disease. I. Explain why the sickle cell allele is more common in the population in countries where the severe form of malaria is present. So sickle cell anemia is a gene mutation in which the amino acid, the glutamic acid, is replaced by valine or valine. In countries where malaria is prevalent, the gene mutates in order to survive, as the Plasmodium parasite causing malaria cannot live in these heterozygous people. As a result, they survive malaria. Selection of the gene is favored in the heterozygous condition, hence carriers are more common in the areas where malaria is common. So this is simply telling us that persons who live in those countries have sickle cell traits, or sickle cell trait is more common in those countries because guess what? It's a type of gene mutation, and as a result, the gene has mutated to favor survival. So it has mutated so these persons living in these areas can survive. And as mentioned, the Plasmodium, which is the parasite that causes malaria, cannot survive in these heterozygous people. They cannot survive in that type of sickle-shaped blood. And because it's a gene mutation, it's in the gene. So once these people populate, then their population would be filled with persons who will carry the gene. And that's why it's common in the area, because it's a mechanism to survive. As it's a gene mutation, it's a survival mechanism. So these persons, their blood or their hemoglobin mutated to enable their survival. So saying this should be okay. And now we would have been at the end of the test. So I would suggest whenever you're at the end of the test, especially if you finish before time, please read over, please check to ensure that you answered all the questions correctly or efficiently or as best to your knowledge as possible. Please ensure that your name is written at the front. So we're at the end of another video. Please remember to like, share, and subscribe. And if you're a returning subscriber, please remember to turn on your notification if you haven't. And I'll see you guys in the next video.